Okay, so good morning, students. I am Dr. Krishnasis Das. I'm currently working as a scientist in Indian Institute of Pulses Research, and here we actually work on biological nitrogen fixation, which is a very important part of pulse crops because you know that pulses are legumes and they have got nodules in their roots and the rhizobium nodule interaction, the rhizobium and plant interaction is very important in fixing atmospheric nitrogen. This is the work we are doing in this institute now. And today, the topic, actually three small topics we will be talking about, like the principles of crop inoculation with microbial agents, then a brief overview of the microbial agents which are being used in agriculture nowadays, and their production constraints and problems associated with their production. So let's start. So as you know, that in the last century, that during this green revolution, chemical fertilizers were used in agriculture. Until now, that indiscriminate use of chemical fertilizer and pesticides is going on. So at the beginning, farmers were very happy because that the response, the crop response towards the chemical fertilizers and pesticides is very drastic, and they got higher yield, increased yield, which sustained our population. But slowly, the chemical fertilizers started displaying their ill effects, which are the leaching out extra nutrients, indiscriminate use of chemical fertilizers, and the pesticides, which are leached out and they are polluting our water basins. This, in terms of time, destroys our microorganisms and friendly insect populations. This also, the luxury consumption of nutrients make the crop susceptible to attack of different diseases and pests. So new kind of disease and pests are coming out. And this also reduces the soil fertility because the imbalanced use of chemical fertilizers actually happens in most of the cases. So they farmers only use nitrogen NPK, but they don't bother about the micronutrients. Now, for proper growth of the plants, we need NPK as well as all the micronutrients like zinc, then calcium, and other micronutrients. But they are not replacing those micronutrients. So the imbalanced fertilizer use reduces the soil fertility, and in the long run, the yield also gets reduced. So a number of intellectuals throughout the world started working on the alternatives to sustain the agriculture. So they found that microbial agents or biofertilizers can help in increasing the yield without causing the damage associated with the chemical fertilizer. So the question arises, what is biofertilizer? Biofertilizer is a substance which contains living microorganisms which, when applied to seed, plant surface or soil, it polarizes the rhizosphere region or the interior of the plant and promotes growth by increasing the supply and availability of primary nutrients to the host. So basically, biofertilizers are living microorganisms which we isolate from the soil, we purify them, then we return them to the rhizosphere zone or root zone where they act as a nutrient supplier or nutrient manager. They actually converts the nutrients to plant available form, which helps in plants uptake and growth. So why biofertilizers are needed? The first and foremost work the microorganisms or biofertilizers do in soil is mobilization of soil nutrients. Now, we have macronutrients as well as micronutrients. And we know that microorganisms are the driving force for this nutrient cycle in soil. So next thing is the synthesis of phytohormones. You must have known about the plant growth promoting rhizobacteria or PGPR. Those microorganisms secrete phytohormones or growth regulators in the root zone or inside the plant, and they help in further growth and development of the plant. The next is protection of plants under stressful conditions. So whenever plant faces this drought stress or heat stress, which is very common in our country, the plants try to force matures, and that's why the yield penalty is there. So yield gets reduced. Now, number of microorganisms are there, mostly the polysaccharide producing microorganisms, which can bind to water particle and they can sustain the plants to a limit so that yield penalty, which is a problem we face under this heat stress or drought stress, that can be substantiated and to some extent compensated. Now the third is these biofertilizers improve soil fertility and enhance the nutrient and water uptake in deficient soils, thereby aiding in better establishment of plants. And finally, defense against plant pathogens and reducing plant diseases or death. Because these microbial biofertilizers, some of them are very good antimicrobial products producing. So these antimicrobial compounds which they produce in the root zone, they can suppress the plant pathogens, and that's why the disease occurrence or the death due to disease may be reduced. So these are the main advantages of biofertilizer. So now this biofertilizer technology, it's not new because the commercial history of biofertilizers began with the launch of nitrogen, which is a actually rhizobium composition by Nobby and Hiltner in 1895. This is the first case. In India also, the first study on legume rhizobium symbiosis was conducted by Dr. N.V. Joshi, and the first commercial production started as early as 1956. That's quite old technology. So now we come to the point that why inoculation of crops with microbial agents is required. Because you may be thinking that microorganisms live in soil, so why we need to inoculate from outside? There are three main reasons. First is the lack of desired population of microbial agents in soil. As you know that if you are going to supply any nitrogen or phosphorus or any microorganisms which drive this particular nutrient cycle, we need the population of the desired microorganisms in the rhizosphere region. Now, often our native soil lacks this kind of desired microbial population. As for example, in 1960s, soybean was introduced in India. So at that time, soybean is a legume crop and it has nodules and the rhizobium infects and it nodulates the plant. But at that time, Indian soil, Indian native soil was lacking this particular rhizobia. And you all know that rhizobia is very crop specific. So at that time, we had to import rhizobium cultures from outside so that our in India soybean crops flourish. Nowadays, it has already well distributed in Indian subcontinent, and there is no need to uh, import any rhizobium culture for soybean. So that is the thing, that lack of desired population. Whenever you grow a crop where this the crop was not grown for a long time, that naturally the rhizobium population gets decreased in the soil. So if you go for crop inoculation, then the crop performance gets enhanced. Next thing is competition among the native microbial species in rhizosphere region. Now, as you know that in rhizosphere region, thousands of species of microorganisms remain there. So those are native microbial species. Whenever you inoculate a crop, your that inoculant should be competent enough to compete with the native microbial species and then establish itself in the rhizosphere region or root zone. Then only you can get the benefit of that microorganism. The third point is also the establishment of desired microbial species in rhizosphere region. Whenever you go for crop inoculation, that establishment rate or the success of establishment of that desired microorganisms in the rhizosphere region gets enhanced. So it is always beneficial for us to go for crop inoculation so that we can ensure the benefit whatever we can get in from that desired microorganisms or microbial inoculation. So there are three main methods of crop inoculation: seed treatment, seedling drip, and soil application. So mostly the easy method with this is that seed treatment method, and then that seedling dip, and then soil application. So the seed treatment, why it is very important? Because the amount of microbial inoculant required is less in seed treatment compared to soil application. Because whenever you want to go for soil application, you have to mix that biofertilizer with compost and then go for application in soil. So the amount of biofertilizer required is quite uh, large in case of soil application. But if you go for seed treatment, that as you can see, that nitrogenous biofertilizer 200 gram per 10 kg seeds. So it is much smaller amount which you can apply in case of seed treatment. And seedling root tip, normally whenever we go for any transplanted this crop like rice or wheat, then you can use this seedling root tip. So among these three types of crop inoculation methods, seed treatment is the best method. And another thing is there, whenever you go for soil application, there is always one problem. Soil is a very heterogeneous medium. So whenever you distribute this microbial agent to the soil, so that seed and that microbial agent, very unlikely that you will get that inoculation perfectly. But whenever you go for seed treatment, we are ensuring that microbial agent is present in the seed coat itself. So whenever that seed will get germinated, that will definitely get inoculated by the microbial agent. So in this way, seed treatment ensures that our crop inoculation goes perfectly. So the seed treatment is a very easy process. I think you all know these things because in case of carrier-based package, you have to just mix it with the required rate of seed, and you can use some medium like water, or somewhere they use jaggery solution to stick that material or powder to the seeds, and then they shade dry it, and then the seeds are ready for sowing. So now we go about the brief overview of the biofertilizers or microbial agents currently being used in agriculture. So in the left-hand side, you can see the 5 different types of microbial agents like Rhizobium, Agitobacter, Azospirillum, PSB, and Mycorrhiza. These are very popular organisms. Among them, most important is Rhizobium because that is related to our nitrogen nutrition. And this Agitobacter, Azospirillum, these are also nitrogen supplier or nitrogen fixers rather than. And these are Rhizobium is a symbiotic organism. So only the legumes are benefited by this rhizobium, but the Agitobacter and Azospirillum is a free living nitrogen fixer. So all other crops and vegetable crops, there these organisms can be used. One by one, we will discuss all these things, and then PSB is there, mycorrhiza is there. Apart from that, nowadays, with increase in microbial technology and this organic farming, we have Acetobacter and other PGPR organisms as well as consortium approach. So to mix 2 or 3 different type of microorganisms to make it a holistic approach. So other different type of cultures are coming out for different aspects, like some are biocontrol agents, some are used in stress condition, and those things, those we'll discuss one by one. So the first thing is rhizobium. uh, this is important because in our input, nitrogen is a main input, and it costs much to farmer. So if in case of legumes mostly, the rhizobium inoculation reduces the nitrogen dose drastically. So rhizobium is the aerobic bacteria which fixes atmospheric nitrogen in legumes symbiotically. Bacteria infect the legume root and form root nodules, within which they reduce the molecular nitrogen into ammonia. This is a great process, biological nitrogen fixation, and for your exam, this topic is quite important. So I request all of you to study this biological nitrogen fixation. topic from any reputed book or these things, and study this process and those genes involved in this PNS process. And it has been estimated that 40 to 250 kg nitrogen per hectare per year is fixed by different legume crops by the microbial activities of rhizobium. Now, this amount varies in legume to legume, and rhizobium are crop specific, so the efficiency of the system varies different legume to legume. And it has been estimated that the rhizobium legume association yield can increase the yield by 10 to 15%. Some has, in some cases, it can increase up to 30%. So this is the legume rhizobium symbiosis process. I guess you are familiar with the process where whenever you go for this crop inoculation, those rhizobium will remain in seed coat or the adjacent area of seed. Now, when the root comes out, this plant root secretes some kind of chemical signal like flavonoids and other Nod factors, which attracts these are actually chemoattractant, and this attract the rhizobium cells towards the root hair. Now this root hair is get infected by this rhizobium cells, and it deforms, and there are different steps as you can see, and this root cell division starts, and then infection thread is formed, and through this infection thread, this rhizobia goes inside the root cells, and then there some phytohormones are secreted like cytokines and all these things. So this the infected cell gets divides, and this forms the nodule. So this is a very complex procedure, and number of factors is involved in this legume rhizobium symbiosis, from plant genotype, then our rhizobium, as well as some environmental factors are also involved, like soil and other factors. So the nodule tissue formation and bacterial differentiation is occur inside the nodule, and then it produces nitrogenase. Now this is another very important topic, this cross inoculation group. You must make a note of this cross inoculation group because it is a very popular question in case of this ARS examination, that what are the crop inoculation group. So these are the main crop inoculation group. You can make one page note on this because it comes in 10 or 5 marks. You can. So now where are the, as I told that the quantity of nitrogen fixed by different rhizobium and legume varies. So this is a comparative table where you can see that different crop inoculation groups, how much nitrogen they fix in per kg per hectare per year time species. So next we come to phosphate solubilizing biofertilizers or PSB. Overall in India, this is the most popular biofertilizer being used because if you go for rhizobium, that is specific to legumes, but in case of PSB, that is not specific to any particular group of crops. It can be used for all the crops, our cereals, millets, all oilseeds, pulses, and vegetables, everything. What basically it does, as you know, that 70% of the phosphatic fertilizer we apply in soil gets fixed based on the pH of the soil. So in calcareous soil, some calcium phosphate and other insoluble materials are created, and this phosphate gets fixed in the soil. So plants cannot use this phosphate for their growth and development. So the phosphate nutrient efficiency, which we call, is very less. Now this group of microorganisms who actually secrete some organic acids which can solubilize this insoluble phosphate, which were earlier plant unavailable form. They convert it to plant available form. So this phosphate nutrient use efficiency gets increased. So in this group of bacteria, different bacillus and pseudomonas bacteria is there, and some fungi are there like Aspergillus, Penicillium, Trichoderma are there. Even some of the yeasts which were prevalent in soil, they can also fix the soil layers, this insoluble rock phosphate. So what is the benefit of using this rock phosphate is that it solubilizes the insoluble phosphate, and our plants growth and this development is enhanced. So this is one small example from a study where you can see this rock phosphate, which is a insoluble form of phosphate, and plant cannot actually readily use this rock phosphate. Whenever you use rock phosphate with microbial cultures, different cultures are there which are actually solubilizing this rock phosphate. You can see the enhancement in number of pods per plant, thousand seed weight, green grain yield. So even BC ratio, these are also increased. So you can see that using PSB is beneficial for plant growth and development. And not only this only phosphate, you may be knowing that nitrogen and phosphorus, these two nutrients are like synergistically related. And so whenever your phosphorus deficiency is there, your nitrogenous activity as well as nodulation activity gets reduced. So these are synergistically related. Whenever you use this rock phosphate solubilizing microorganisms or PSB with rhizobia, we have got better results. Now the third part, this BAM, earlier it was very popular in this soil, but nowadays other microorganisms have come, so definitely their popularity has reduced. So BAM is a fungus which colonizes the plant root system and increases the growth and yield of the crop. What it does in soil, actually, it this is a fungi which creates a mat around the root of plant, and this increases the surface area of the root, which then helps plants to acquire nutrients as well as water and macro and micronutrients from soil. And this increased surface area helps plant to acquire nutrients from greater distances. So the whenever in drought condition or in stress condition or in soils where nutrient deficient soil is there this vesicular vascular mycorrhiza is very important to plants. I request all the students to go in book and you study about the different type of vascular mycorrhiza. That is, the classification is there, so you can make one small note on different type of VAM available and that classification of vascular mycorrhiza. Now, this is one study in our institute where you can see that in case of this control, see the root growth, and then whenever we apply the BAM and PSB and BAM with PSB, you can see the extent of root growth and that volume gets increased as well as the plant growth and vigor gets increased. So definitely this BAM helps plants to extract nutrients from soil pretty extensively. Now, as I said, like after this nutrient mobilization, new group of microorganisms coming out. So which biogens are specific for abiotic stress management. Now this group of organisms in total we call plant growth promoting organisms. Actually, these there are different methods where this plant growth promoting rhizobacteria or organisms help plant. Like first of all, there are producers of phytohormones or phytoregulators like cytokinins or IA and other metabolites which actually alter the root morphology and their transporter activity. So the higher root growth helps plants to acquire nutrients and as well as the cytokinins and this auxins, which helps for higher root growth, which help in above ground development and then increases the yield. Now, next thing is the antioxidants. Whenever drought stress is there, that reactive oxygen species are generated, which are actually harmful for plant growth and development. Now, these microorganisms excrete some kind of antioxidants and mechanism is there like superoxide dismutase and all these things, which helps to reduce this reactive oxygen species and which help plant to survive in drought stress. Then there are SEC deaminase. We will study about this SEC deaminase, how it helps plants for compensate in drought stress. Then there are salt, as I said earlier, the polysaccharide producing microorganisms are there, which help in enhancing that sodium translocation and sodium pump in root zone of the plant, which then further help plant to survive in high salt, the saline soils and the sodic soils. So now we briefly will discuss this ACC deaminase activity because this is a well-studied phenomena. Like this enzyme, 1-aminocyclopropane, 1-carboxylate deaminase. This is a enzyme which flips the ACC compound into alpha-ketobutyrate and ammonia. Now, why this is important? ACC is a precursor of ethylene molecule. Now, whenever plants are in stress condition, this drought stress is there, this ethylene is produced inside the plant and this forces maturity. So plants get mature quickly, senescence is there. So the yield penalty is there because plant actually tries to complete its life cycle before the drought stress is there and terminal stress is there. Now, this bacterial isolates what it does that micro ACC deaminase cleaves this ACC compound, which is the precursor of ethylene molecule, into two different molecules, which is alpha ketobutyrate and ammonia. Now, ammonia is again used as nitrogen source, and that ethylene uh, production gets reduced, which helps in longer root growth and better survivability of these plants under drought stress. In the photographs also, you can see the control and then whenever the ACC is, the precursor molecule is there, the root growth is stunted, root growth is there, it gets reduced. And then whenever we use the ACC with bacteria which produces this ACC deaminase enzyme, the root growth is compared to control and even better in stress condition because it cleans the ACC and provides the plants the necessary support under stress condition. So here also in one study, we have found that in control plant, you can see the growth and then in 30% water holding capacity, the plants get dried up due to lack of moisture in soil. Now, whenever we use this isolate which produces ACC deaminase enzyme, you can see the plant survivability gets better and the plant survives and it produces seed or we can get some yield. So the total yield loss is prevented and the plant growth is better in case whenever we apply this ACC deaminase enzyme producing microorganism. But now these microorganisms actually don't act single-handedly. They are used in a consortium format. Now here also in another study, you can see the how microorganisms which produces ACC deaminase increases the root length, root volume, and root dry weight in plants in stress condition. So definitely under stress, this kind of microorganisms help to enhance the growth and development of plants. Now, as I said, like in soil, actually, you cannot say that one single microorganism is working alone. That ecology provides that different microorganisms interact with your desired microorganisms or microbial agent. So definitely, this is one thing that some competency should be there, that your microorganisms, whichever you are applying to seed or soil, gets established in the soil. So this co-inoculation study is very important whenever you are applying any PGPR or any rhizobium or PSP. Some co-inoculation study should be there. This is not in farmers level. This is actually in researchers level. The researchers, whoever is developing this kind of bioinoculant, they use some co-inoculation studies. In this study, we try to see that if rhizobium and ACC deaminase activity producing microorganisms, if we co-inoculate, if that inoculation is harmful towards rhizobium or not. In this study, we found that these two microorganisms are quite compatible, and you can use these two microorganisms simultaneously in seed. So apart from these microorganisms, actually there are other microorganisms like Agitobacter, Azospirillum, free-living microorganisms are there. What is the difference between this rhizobium, which is symbiotic microorganism, and this free-living nitrogen fixer? Is actually rhizobiums are specific to legume crops, and this Agitobacter, Azospirillum, you can use in other crops like cereals and vegetables also. Now, the amount of nitrogen fixed obviously gets different, because in case of rhizobium, you get a specialized organ called nodule. So, there that oxygen, as you know, that nitrogenous enzyme, which is important for fixing atmospheric nitrogen, is very sensitive to atmospheric oxygen. So in legume, one specialized this pigment, that is leg hemoglobin, is there, which regulates the oxygen. So the oxygen concentration gets regulated, and that's why the amount of nitrogen fixation is higher in case of rhizobium legume symbiosis. But in case of Agitobacter and Azospirillum, these are free-living organisms, and obviously within cell they have some kind of mechanism which protects their nitrogenase. But since there is no specialized organ like nodule or any form, so that's why the amount of fixed nitrogen is comparatively low in case of free-living nitrogen fixers than this rhizobium culture. So apart from that, other PGPR specialized microorganisms are coming up. Obviously, I guess separate lecture will be there for this biopesticide and biocontrol microorganisms, which is another important aspect, because biofertilizer, two different aspects are there. One is this nutrient aspect and phytoregulators aspects, and then another is bio control agents or biopesticides, where we deal with microorganisms, including antimicrobial compounds, which actually prevent those pathogenic microorganisms or kill them, control them, suppress them. And how this kind of microorganisms are used, obviously there must be some separate lecture to discuss this antimicrobial compound. So next, we come to the production aspect, because once you have identified the microorganism, you have isolated from soil or any other ecological source, and then you grow it in culture, in laboratory, in petri plates, in or in media, in broth culture. As you can see, the broth culture is there. But then this is, if you use this culture as such in the field, this cannot establish itself perfectly, or there are some other problems like farmers, though they don't use it instantly, they have to store it for some time, like 1 month or 2 months, or whenever you go for commercialization, definitely the shelf life has to be there. Now, if we go for this batch culture, what we call in task and these things, that nutrient is limited there. So you must have learned that the microbial growth curve, that log phase, stationary phase, and death phase. Whenever the nutrient is not supplied, if we don't add any kind of like preservatives or some kind of protective mechanism should be there to maintain that stationary phase of this bacterial growth curve. If you don't maintain this stationary phase, that obviously with limited resources, your microorganisms will go into death phase. And once it goes into death phase, definitely your efficiency of the microbial inoculant will be reduced. This problem has been solved by microbial formulations. So what is formulation? Formulation is actually some kind of mechanism to compensate this death or this arrange this stationary phase in your culture. So there are two main different kind of formulations in market. One is powder based or granular based carrier based microorganism this formulation, and another is liquid formulation. So first, we will discuss about the carrier-based microbial formulations, which is very popular, and this is the first initial phases. We used to produce this carrier-based. It's very easy to produce, and we can discuss about the properties of a good carrier material for seed inoculation. Now, the carrier material is very important because this absorbs your broth or microbial broth, and this carries your microbial cells or rather than viable cells to your inoculation site, that is root. So definitely some criteria are there for the better carrier, which are the good carrier material. So first and foremost, that carrier material has to be non-toxic to inoculant bacterial strain, because if that carrier material is toxic to bacterial strain, definitely the viable plant count and cell count will be greatly reduced, and that culture is will the shelf life of culture will be drastically reduced. And second is good moisture absorption capacity. Actually, whenever you go for production of this carrier-based or powder-based formulation of microbial agents, that we recommend that 40% moisture holding capacity should be compensated by this culture, because like 100-gram culture should absorb 40 ml of liquid broth. This is the kind of thumb rule. So good moisture absorption capacity should be there. So that's why you must be remembered that some of the materials which are powdered gets that volume gets increased, and they are very good moisture absorber. Like perlite is there, and very common our charcoal, that peat. It's also a good moisture absorption capacity is there compared to vermiculite or any other synthetic clay materials. We will discuss also. Then third thing is that easy to process and free of lump forming material, because whenever this natural materials, natural carrier materials are used, it should be processed, because from lump you cannot use that. You have to grind it to make it fine powder, and then only its moisture absorption capacity will be increased, and then your powder will be free flowing, which is for seed treatment. It will be very easy to mix with water or jaggery to make this seed inoculation uniform. If that lumps will be there, then it will not be that much user friendly, as well as the efficiency will get affected. Now the fourth point is that carrier material should be easy to sterilize by autoclaving, or nowadays gamma irradiation is there. Now this sterilization process is very important, because that you don't want to contaminate your desired microbial agent or microbial culture with other native microbial populations which resides in this carrier material. uh, Naturally, obviously these are natural material, these microorganisms will be there, so we have to sterilize this autoclave through autoclaving this material so that you kill those native microorganisms, then you inoculate with desired microorganisms to make your biofertilizer. Fifth point, should be available in adequate amounts. Obviously, that if you are going for commercial production, then the production unit should has to be supplied with sufficient quantity of carrier material. Inexpensive, obviously, that carrier material. In next slide, we will discuss different type of carrier material used, and definitely some of the carrier materials like silica gel and all these things are very expensive, so it increases the cost of input and the cost of biofertilizer. On the other hand, like very popular thing, this charcoal, the lignite, peat, and this compost, these are very inexpensive. So definitely your cost of production gets reduced, and farmers can afford this low cost biofertilizers. So the next point is good as addition to seeds. Obviously, you are going to coat seed with your material. So definitely that adhesion to seed is very important because this material will carry the microorganisms. It has to attach to itself to its seed coat so that whenever the seed gets germinated, your microorganisms naturally infect those root. Next thing is pH buffering capacity. Why it is important? Because we know that the microorganisms are living organisms and they have a proper pH range where they can flourish. If you use any kind of carrier material which is too acidic or too basic, it will affect the viability of the microorganisms. And so next point is non-toxic to plant and environment. Obviously, you are going to apply it in soil or seed coat. Definitely, that carrier material has to be biodegradable material. Now, a diverse array of soil materials are used as carrier materials. The most popular material are in the first line, like peat, charcoal. In some area where volcanic fumes is available, they use the volcanic ash or this clay. This material, obviously, different material has its pros and cons. The peat and charcoal, these are very easy to get. These are very inexpensive and very easy to work with. But the problem with the peat and charcoal is the contamination is much higher in case of these things, and also affect the performance of the microorganisms. Then there are organic materials like compost and different plant byproducts like straw and all these things. Some of the biofertilizers work very well with this compost and plant byproducts like straw or husk and all these things because those microbial inoculants which are used for this compost making. So or this soil application. There you can use this carrier material, it works perfectly fine. But again, then contamination is one of the most problems with which is related to this carrier materials, powder-based carrier materials. Then there are inert materials, perlite, vermiculite, bentonite, kaolinite, clays, and silicates. These are inert materials. Obviously, they don't interact with your microorganisms. So, this performance of these microorganisms get enhanced. And these inert materials are very much biodegradable, but the cost of production gets increased. So, definitely based on the economy or economy of the microbial biofertilizer production, different type of carrier based microorganisms carriers are used. Now we have to understand that this technology was initially used and it is still popular, carrier based microorganisms biofertilizers, but this has some disadvantages like availability of good quality carrier material is limited. Like whenever you use peat, charcoal, or all these things, these are available, but uniformity of the product, it's a difficulty, because you cannot this assure that the quality of peat or charcoal will always be same. The composition changes with batch to batch. So definitely, your whenever you are going for commercialization or commercial scale this biofertilizer production, it may affect the uniformity of the such a material. So efficiency also gets affected. Next is mining of this peat, charcoal, or all these carrier materials, processing, grinding, sterilization. All these things are labor-intensive process. Next is survival of microorganisms or rhizobia on peat or charcoal is limited. So you have to add some kind of media or some system or some kind of nutrient so that the survival rate of these microorganisms on this carrier material gets enhanced. Now the storage is also another problem. It has to be stored in low temperature because carrier materials get heated very easily. So the buffering capacity is less, and that's why obviously the shelf life of this carrier-based inoculum is shorter. So what is the way out? So nowadays, that liquid inoculants has simplified the production process, and there is no need to prepare and amend a carrier, and then application to seed or field is much easier. So definitely, liquid inoculants and liquid bioformulations have come out with big time. So what are the advantages of liquid biofertilizer? Definitely, first and foremost comes that contamination. In case of carrier materials, these are natural materials, so and these are powder. So whenever you autoclave them, often it has been seen that the heat doesn't penetrate uniformly, so that material is not sterilized properly. So this becomes the source of contamination. And as you see, microorganisms are very tiny microorganisms. If some lumps are there in your carrier material, microorganisms, petty microorganisms can survive in those lumps, and at later stage it will contaminate your the whole batch. You will get contaminated. And second thing is their identification is of contamination, that your culture has got contaminated. It's very much difficult in case of carrier based or biofertilizer. But in case of liquid biofertilizer, liquid media, you can autoclave it uniformly, easily, and whenever it gets contamination, somehow if it gets contaminated, you can easily identify that cottony growth is there or any other different kind of microorganism pigmentation is there. Then you can identify that your culture has got contaminated. Next is longer shelf life, and it can be stored in high temperature. High temperature, that means our room temperature in case of liquid biofertilizer. Liquid biofertilizer, as whatever we are producing nowadays, it can be stored up to 1 year. But in case of carrier-based biofertilizer, it is only 5 to 6 months. So next is the higher microbial population. Obviously, whenever you grow microorganisms liquid broth culture, you provide high nutrient dense media, so the cell population is much higher in case of liquid biofertilizer. As you can see, that cell population can go up to 10 to the power 9 cells per ml can be maintained for its entire shelf life. So whenever you apply, even if you dilute that 10 times or 100 times, you get enough amount of microbial load on your seeds, which is very important for establishment of microbial cultures in this root zone or on root. And then much simpler application because it is liquid, it is very easy to mix with the soil, mix with your seed material and seed coating, and the dosage can be perfectly used, and lower dose of liquid biofertilizer can work as compared to your carrier-based material. So it is quick and easily applied. Quality control protocols and amelioration is very easy because as I said, like identification of contamination and quality control issues can be solved very easily in case of liquid biofertilizers. And then obviously the higher export potential, weight and volume gets reduced in case of liquid biofertilizers. You can produce a dense, high concentrated liquid biofertilizer, and then in field you can mix it with some water or any sugar solution and use it. So definitely that export potential is higher in case of liquid biofertilizer. Now, the fertilizer control order has some efficiency requirements for different strains which are being used nowadays. So, if you concentrate like viable cell count, this is the most important point for any carrier-based or liquid-based formulations. The CFU minimum has to be 5 × 10⁷ cells per gram of powder or 10⁸ cells per ml of liquid. That is your culture material that has to be there. Contamination, obviously, there should not be any contamination. pH should be near neutral, 6.5 to 7.5. That is neutral pH, which is helpful for survival of microorganisms also, and this is not detrimental to plants also. Particle size, in case of carrier-based material, some particle size sieves are there, and moisture holding capacity, which I said, like 30 to 40% should be there to moisture holding capacity of the carrier material. Now, for different organisms like Rhizobium, Azotobacter, Azospirillum, PSB, some different small criteria are there which are required for whenever you are going for commercial level microbial agent production or microbial unit. So, these are the points actually should show effective nodulation all the species listed on the packet. Obviously, this is for Rhizobium because Rhizobium is a crop specific. So whenever you are using, you must ensure that the crop species or whatever crop you are using, the Rhizobium is specific for that crop. Now, mycorrhiza, actually, previously, this some kind of specifications for mycorrhiza was there. Obviously, nowadays, the mycorrhiza population gets the use and popularity of mycorrhiza is getting reduced. Now, this biofertilizer technology or microbial inoculant technology is quite old in our country. And after 50, 60 years of research and all these things, we have all the cultures. But definitely the popularity of all these cultures are not at that level which we desire. Because farmers are still dependent on chemical fertilizer, rather indiscriminate use of chemical fertilizer rather than supplementary use of these microbial agents. So what may be the problems in dissemination as well as production of microbial agents in our country? First and foremost, the quality control issue for microbial formulations are there. So studies are being made to define the minimum standards and testing protocols for all biological and organic inputs. Like chemical fertilizer, that purity check and all these things are very easy. But in case of biological, this is a diverse process and it's a living organism. So proper guideline has to be there in case of this biofertilizer production. And the proper shelf life has to be ascertained, because in case of chemical fertilizer, the shelf life is quite naturally it's higher, but in case of biological, shelf life is very important, because if the culture is dead during the storage process, then it is of no use to farmers. Different biofertilizer, 5 biofertilizers, 3 organic fertilizers are covered under FCO, and efforts are underway to define the standards and quality methods for trichoderma and pseudomonas, mixed culture or consortiums. ज़्यादा होगा या फिर ऑफिस में दे ऊपर ऑफिस हाँ जी किसी सॉरी फॉर द इंटरप्शन So, what are the precautions one should take for using those biofertilizers? First of all, biofertilizer packets need to be stored in a cool, dry place and away from direct sunlight and heat. Obviously, it is a living organism. Direct sunlight and heat may reduce the viable cell count. Right combination of biofertilizers have to be used, which I said, like obviously this commercial producers, they study these combination studies, and they know that PHB can be used with rhizobium and all these things. So those data are there with them. So they ascertain that the combination of biofertilizers can be used. As rhizobium is crop specific, one should use for the specified crops only, and other chemicals, especially these chemical fertilizers and these pesticides cannot be mixed with biofertilizers. Obviously, there is one process like seed coating with different carbendazim and other microbial agents in single seed. So you must be knowing that FIR is there. First is fungicide, whatever fungicide is there, you coat it the seeds with this fungicide, and then you shed dry it, and then go for I, that is insecticide. Whatever insecticide you want to go for seed coating, you can coat the seed with insecticide, and then again shed dry it, and then lastly you can go for rhizobium, that is living organism that should be in the outer layer. So FIR, you remember this keywords. This is the sequence of sheet coating with different chemicals and with living microorganisms. The next is the packet has to be used before its expiry of only for the specified crop and by the recommended method of application. Dose of application is also important because it's living organisms, and the amount minimum cell numbers has to be assured and seeds for better crop performance or better crop establishment. It is highly beneficial to use biofertilizers along with organic manures because it is often seen that organic manure or this organic matter or FYM that has got this carbon source and nutrient source, which helps the biofertilizer microorganisms to grow luxuriously in the rhizosphere region. If they grow luxuriously, definitely the crop establishment or this microbial establishment will be higher. So we have already discussed that the advantages of biofertilizer use. The nitrogen biofertilizers can provide 25 to 30% chemical fertilizer equivalent nitrogen. You can reduce 25% of nitrogen through application of this biofertilizers. PSB obviously 12 to 20 kg P2O5 per hectare per season can solubilize this microorganism. Mycorrhiza can provide adequate phosphorus, micronutrients, and helping increase water absorption. And consortium nowadays, actually, consortium approach is coming up, and it is better. Consortium is mixing of 2 or 3 compatible microorganisms with different nutrient mobilizers or all these things, which can be used as a combination in soil or in seed. So definitely in one at a go, you don't need to coat for phosphorus or nitrogen separately. You get all 3 nutrient benefit in one single consortium microorganism biofertilizer. So obviously this consortium approach is better. In another way also, the consortium gives the better chance of establishment in your root zone. Keep soil biologically active. Obviously, this soil organic matter gets increased and microbial population, the metabolites get secreted, and the soil aggregation and soil biological part gets enhanced, and which in turn helps soil health maintenance and sustain our agriculture. So finally, we come to the biofertilizer production scenario in India. As I said, like biofertilizer production in India has increased from mere 25,000 metric ton in the year 2008-9 to 1,21,000 metric ton in the year 2017-18. This is the NCOF data, and you can see this is actually increased mostly in case of carrier-based biofertilizers. Liquid biofertilizers came out late, and now it is getting popularity. among the farmers. It has been estimated that in India, the money spent on biofertilizers and biopesticides is around 1.5 billion. That is a huge market, and obviously, that the training of organic farming and environmental awareness, the use of biofertilizer is expected to increase in the coming years. Till now, carrier-based solid biofertilizers are more popular in India, and among all the carrier-based microorganisms or biofertilizers, the phosphate solubilizing bacteria is the most widely produced biofertilizer, and closely followed by azotobacter and azospirillum. And naturally, due to its improved quality and longer shelf life, liquid biofertilizers are rapidly gaining popularity among farmers. And on all the liquid biofertilizers, PSB and NPK biofertilizers, the consortium are most popular. In zone-wise production, if you see that the South zone comprises more than 60% of biofertilizer production in India. Our southern states like 7 states, Karnataka, Tamil Nadu, Maharashtra, Kerala, Madhya Pradesh, Gujarat, and Andhra Pradesh account for more than 80% of total biofertilizer production. You can see in the pie chart and the state-wise contribution in the production of carrier-based biofertilizer in India during 1718, that Karnataka ranks the first in production of carrier-based formulation, followed by Tamil Nadu and Maharashtra. Whereas in case of liquid formulation, liquid biofertilizer production, Gujarat is leading in this liquid biofertilizer production, followed by Maharashtra. The 39% of liquid biofertilizer production occurs in Gujarat. So, then we come to that poor accessibility, which I was talking about, that popularity of biofertilizers are not at that level which we expected, because some points are there like inconsistent response. Carrier-based biofertilizers, the consistency or uniformity of material, plus that viable cell count is very difficult to maintain, as well as there is no proper regulatory mechanism. That's why the inconsistent response or performance in the farmers field discourages farmers for using this biofertilizer. Poor quality of carrier-based products I have already discussed, and that sensitivity to temperature and short shelf life, because in our country the diverse ecological ranges are there. Somewhere we can expect in western part the temperature in summer goes up to 45 to 50 degrees centigrade. Obviously, this living microorganisms, they cannot survive, or rather, it is very difficult for them to survive in those high temperature days. So their storage facility should be there. Some kind of mechanism should be there so that farmers get quality material. Non-compatibility with chemical seed dresser and fertilizers. Obviously, this segment needs more research in case of our post-harvest technologies and machineries. They can take care of these things like biofertilizer compatible machineries should be there. Poor organic carbon in soil. You all know that Indian soil is naturally very poor in organic carbon, and this organic carbon or organic material is very important in case of establishment of our this microbial agents, because this work as nutrient for those microbial cells. And dependence of supply on government system. Obviously, government system has come up with biofertilizer and all the things with training organic culture and organic materials. Obviously, these things will get changed in future. Some corrective measures needed at production stage. Obviously, that poor quality and under sterilized carrier materials are biggest concern. We can manage this thing. Let's replace lignite charcoal with other carriers having high moisture potential, such as vermiculite, silica, quartz, and inert material mineral powders enriched with synthetic water holding gels. But this kind of technology is with us, but this increases the cost of production. And then again, this comes if the cost of production gets higher, then farmers get discouraged, or some of them will not be able to afford this kind of biofertilizer. Although the biofertilizer price has been quite in check compared to chemical fertilizer. All carriers must be sterilized. Product handling process must be automated. With industrialization, the biofertilizer production facilities are being automated, and that automation helps in products with free of contamination, and product quality gets increased. So with this, we come to almost end of the presentation. Like efforts are needed to replace existing carrier-based system to liquid system, because liquid systems are easier to produce, and automation, 100% automation is possible. And as I discussed, product can be 100% contamination free. Monitoring will be easy, and higher shelf life can be ascertained. It will be temperature tolerant, and the application of liquid biofertilizer through machineries and sprayers is very easier rather than carrier-based materials. And finally, the introduction of mixed inoculants or consortia, as I discussed, like consortium approach is better than single inoculants. Many producers have launched mixed inoculants like Azotobacter, Azospirillum, PSB, NPK biofertilizers are there. So, in coming years, we are going to see more increase in this consortium approach. So, finally, it is evident that whatever chemical fertilizer or whatever soil reserve is there, you can tap into that reserve through microbial intervention. Microbial cells or microbial inoculants can solubilize or mobilize these nutrients which are already in soil, or but it is in fixed manner. So microorganisms can mobilize these things, and provide to plants, which finally increases the plant growth and productivity. And side by side, it also helps in maintaining the soil health and sustain soil fertility. So definitely in coming years, this is going to be a big news, and will come up in big space. And for the base, but regulatory mechanisms has to come. Any farmers should get the quality microorganism or quality biofertilizer, so that they can use it, get rely on it, and the popularity of microbial biofertilizer. With this, we come to an end. Thank you for patient hearing, and if you have any questions, you can ask.